students, today we will learn about the fungal and algal agents for food-borne illnesses. As you know, food safety is a remarkable interest area and it has extensive public health implications. Food safety alludes to all those hazards which creates the food unsafe for health. The entire food should be harmless, devoid of contamination, and the spoilage and decay of the foodstuff and the farm products through a mixture of toxin producing molds and algae is of major concern. Natural borne toxins generated by microorganism could be a source of epidemic of various infections. Among the microorganism, fungal and algal toxins are of significance because of their universal dissemination. Fungal toxins are responsible for poor health of human and animals because consumption of infected product causes ill effect on the consumer health. Milk, being one of a rich source of nutrient, is a good medium for the growth and propagation of many harmful microorganisms, and therefore it is highly susceptible product from the general healthiness. As I have already mentioned that milk, being a rich source, can harbor a number of microbial pathogens, and these gain access during different stages during the milk production and processing steps. These include milking, handling, transportation and processing. Also, the equipments used for milking, filtering, cooling, storing and dispensing of milk are some of the key sources of microbial contamination. Also, one of a major source is the infected cattle or animal, and also the workers who may be handling the different steps of the process. There are also tertiary microbial contamination as well as recontamination of milk after it is being processed because of unhygienic conditions of the storage of the milk. Contamination of water and milk can introduce many pathogens, and among the pathogens, fungi and algae are two important agents. Mycotoxins, that is the toxins produced by the fungi, can enter the food chain by various means which may be direct as well as indirect. Flies may also provide as a vector for the spread of the diseases. Toxigenic fungi produce several kinds of poisonous secondary metabolites, but all molds are not poisonous and not all the secondary metabolites produced by the fungi are lethal in nature. The known genera of mycotoxigenic molds are Penicillium, Fusarium, Aspergillus, Stachybotrys, Claviceps and Alternaria. Among the toxigenic fungi, there are four broad groups. First, pathogens of plant like Alternaria alternata, Fusarium graminarium. Second, molds that mature plus generate fungal toxin on stressed out plants like Fusarium moniliforme and Aspergillus flavus. Third, fungi that originally inhabit the plant life as well as enhance the raw material vulnerability to spoilage following crop harvesting such as Aspergillus flavus. And the fourth group is fungal species which could establish on top of the soil or putrefying plant substances which appear onto the budding kernels inside the field. For example, Aspergillus ochreus and Penicillium verrucosum. Now, let us know what is mycotoxins. The term mycotoxin was first used in the year 1961. The disease was related to meal of peanut, which included in the diet infected by means of a toxin formed as a result of filamentous fungi Aspergillus flavus. According to Food and Agriculture FAO, a minimum of 25% of the world's food crop are influenced through fungal growth. Food products are contaminated with mycotoxins during, before and after harvesting of the crop. The mycotoxins predictable inside the food diverge from countryside with respect to different crops, agricultural practices as well as environmental circumstances. So, let us know what are mycotoxins. Mycotoxins are low molecular weight compound. Chemically, they vary from simple C4 compound to complex substances. Food poisoning caused by the ingestion of mycotoxin is known as mycotoxicosis. Few mycotoxins have allergic properties also, and they may cause autoimmune illnesses. Others may cause teratogenic, carcinogenic and mutagenic impact on the health of human beings. The majority of the mycotoxins exert influence on the blood, skin, kidney and central nervous system. Detrimental consequences can vary from nausea to weight loss and a variety of tumor, and also it may lead to fatality. A variety of mycotoxins are significant in food items. We will briefly see some of the major mycotoxins, what are the feed or the food on which they are present and what are their toxic effect on the health of the consumer. The first group of mycotoxins are Aflatoxins. These are produced by Aspergillus flavus, Aspergillus parasiticus and some Penicillium species. A wide variety of food items may be a source of the growth of these fungi. These may include cereals, pulses, crops like cassava, cottonseed, peas, sweet potato, unpasteurized sheep, egg, beef, milk, etc. And it is toxic to a wide variety of consumers, include animal, chicken, dog, human being, pigs, etc. Okra toxin A is another mycotoxin which is produced by a diverse group of fungi like Aspergillus, Penicillium species, etc. And the substrate on which it is found also include cereals, millets, legumes, eggs, fruits, coffee, cheese, etc. And it has been found to be toxic to rat, chicks, duckling and trout. Talins, which is produced by Penicillium species, different species of Penicillium and Aspergillus, Aspergillus clavatus, Aspergillus tereus are producing tallins. Apple and its products such as fruit extract, vinegar, food stuff for the babies are the major substrate on which this is present, and it has been found to be toxic on a range of animals, mice, rat, cat, shrimps, etc. Penicillic acid, which is produced by Penicillium mauritius, Penicillium cyclopeum, Penicillium madriti, Aspergillus quercinus, Aspergillus sulphureus, etc., is present on dried beans and tobacco, and it has been found to be toxic to rats. Roquefortin is another mycotoxin which is present on blue cheese, and it is produced by Penicillium roqueforti. Fusarium toxins are also produced, which are present in egg, bovine milk, corn flour, dehydrated fig, maize, etc., and it has been found to be toxic to pig along with chicken. Major toxigenic fungi and their toxins produced which are harmful to cattle are some of the fungi like Aspergillus, Penicillium, Fusarium, Claviceps, etc. are known to produce mycotoxin. The toxins produced by Aspergillus are Aflatoxins, primarily Okra toxins, Fumitoxins, Gliotoxins, etc. The Penicillium produced citrinins, penicillic acid, roquefortin, mycophenolic acid, PR toxin, etc. The fungus Fusarium produces zearalenone, monoliformins, fumonisins, fusaric acid, fusarium C, etc. The claviceps fungus is famous to produce an ergot alkaloid inside the seed grains of small grain grasses and sorghums. One of the toxin which was first identified was aflatoxins, which were identified in 1960s when 1 lakh turkey population expired following consumption of infected aflatoxin peanut meal in United Kingdom. This is a hepatotoxin and carcinogenic in many species. The 3 most significant Aspergillus species which generate aflatoxins are Aspergillus nomeus, Aspergillus parasiticus and Aspergillus flavus. The favorable conditions that help in the synthesis or these toxins are warm temperature and a humid atmosphere, that is warm temperature ranging in 13 to 35 degrees Celsius and a humidity of water activity more than 0.93 is ideal for the production of aflatoxins by the Aspergillus. Mycotoxin of Aspergillus are a chain of 18 recognized bisulfurane polycyclic compound that fluoresce vigorously under UV light. Aflatoxin B1 along with B2 generate blue fluorescence, while G1 as well as G2 generate green fluorescence. Aflatoxicosis is demonstrated with pain in abdomen, vomiting, coma, convulsions, heart and liver diseases, kidney fattiness and death with cerebral edema. Aflatoxin B1 in the dairy products changes to a metabolite aflatoxin M1, and this has been identified in pasteurized milk, powdered milk and ultra high treated milk along with supplementary milk based products. According to FDA regulations, feed or food comprising aflatoxins at regulatory levels of 20 part per billion of AFB1 are preferred for human being foodstuff. Intensity up to 300 parts per billion is acceptable for particular supplies, and 0.5 ppb of AFM1 in milk samples. Mycotoxins of penicillin are produced by some species of fungi like Penicillium expansum, Penicillium verrucosum, Penicillium hirsutum, Penicillium roqueforti, Penicillium crustosum, Penicillium viridectum, Penicillium chrysogenum, Penicillium citrinum, etc. These produce a number of mycotoxins like Okra toxin, penicillic acid, citrinin, roquefortins, cyclopyrazonic acids, talins, etc. Okra toxins, which is abbreviated as OTA, are first discovered in South Africa in 1965. Foodstuff crops such as corn, barley, rice, flour, rye, oat, peas, green coffee beans and pancake mix are reported to have okra toxin. Humans are exposed to okra toxin either directly via the food chain or indirectly via the feed chain. These shows immunotoxic, genotoxic, neurotoxic and teratogenic and embryotoxic effects. It also exerts an immunosuppressive effect. Now we come to mycotoxins of Fusarium. Fusarium, the fungus, develops an array of diverse phycotoxic compounds, for example, fumonisins, fusaric acid, iniatin, buvaricin, zearalenone, monaliformin and other toxins. Fumonisins are a collection of non-fluorescent mycotoxins and they were discovered first in 1988. It is formed by several species of Fusarium, particularly Fusarium nimgemae, etc. This particular mycotoxin is present in a number of agricultural foodstuffs such as corn, corn flour, dried figs, dried millet, maize fractions, bovine milk, medicinal plants, herbal tea, etc. According to WHO report, intake of fumonisin B1 by human being is about 80 nanogram per kg per day. And according to International Agency for Research on Cancer, these toxins are identified as possible carcinogen. It is also found to be immunosuppressive in nature. In case of human being, everyday consumption of fumonisin must be lower than 1 microgram per kg of the body weight per day. Are also known to produce mycotoxins of different chemical nature. Some of the common fungi are Alternaria acrimonium, Claviceps chaetomium, Cladosporium, Myrothecium, Rhizopus and Rhizoctonia. Majority of these molds occur in animal feed and their consequence to food protection possibly is negligible. Of special interest is Claviceps purpurea, which is also known as the ergot mold. It is the causative agent of ergotism and numerous toxins of Alternaria have also been characterized. So, the question is how to prevent and control the occurrence of these mycotoxins in the various foodstuff. The first and the foremost condition required is to employ good agronomic practices during the pre and post harvest period. And then it is essential to utilize genetically resistant hybrids which are resistant to the growth of the fungal attack. And secondly, by managing the moisture and temperature of the stored food product. Chemical treatment with compounds like ammonia, sodium thiosulfide, bases and ozone also helps. Biocontrol method using Bacillus species, lactic acid bacteria and propionic acid bacteria is also found to be effective in reducing the incidence. The other important thing is to pasteurize and follow the HACCP practices and good manufacturing practices. Sanitation and milk product preservation by adopting refrigeration is an important step towards preventing the occurrence of mycotoxins. The examination of cattle and exclusion of the diseased animals is a preventive measure. Scheduled therapeutic examination of entire milk handler will also assist in reducing the incidence of mycotoxin present in the various food items. There is another group of organisms which are also known to produce toxins in addition to fungi, and this group is known as the algae. The algae are source of different type of toxins, especially the harmful algal blooms and members of Bacillariophyta or diatoms and dinoflagellates, which has accumulated in the aquatic food chain. It is essential to compare the marine toxin, aggregation of cyanobacterial toxin in the food chain and subsequent significance of it to human being along with environment, and there is very little knowledge about it. Now let us see what are the important toxic algal dinoflagellate toxins. Dinoflagellate is a group of eukaryotic algae in the aquatic ecosystem. Foremost casual representative of harmful algal blooms, these toxins are functionally and structurally different and several illustrate exclusive biological activities. Different seafood poisoning syndromes occurred by ingestion of dinoflagellate toxins and such contaminated seafoods. Neurotoxic shellfish poisoning, which is abbreviated as NSP, paralytic shellfish poisoning PSP, Sigargutra fish poisoning CFP, Dyric shellfish poisoning and amnesic shellfish poisoning are some of the examples. Functionally, these could be designated as hepatotoxins, neurotoxins, and it depends on their clinical symptoms. Now we will learn about some important toxigenic algae. The first in this group are dinoflagellate toxins. Dinoflagellates are eukaryotic algae present in the aquatic ecosystem. They are responsible for harmful algal blooms. Their toxins are functionally and structurally different from other compounds and they show exclusive biological activities. Different seafood poisoning syndromes occur following ingestion of dinoflagellate toxins contaminated seafood. Neurotoxic shellfish poisoning, paralytic shellfish poisoning, Sigurata fish poisoning, Dyric shellfish poisoning and amnesic shellfish poisoning are some example. Most of these toxins are hepatotoxin, neurotoxin, and it depends on their clinical symptoms. The toxins produced by a specific group of algae which are known as cyanobacterial toxins. These cyanobacteria are able to form harmful algal blooms in different aquatic habitats. In fresh water, these blooms are developed as a result of growth of Anabaena species, Microcystis species and Oscillatoria species. And by their growth, they influence the safety of a variety of seafood and the nutritional supplements derived from these seafoods. They may form hazardous bioactive compounds, and these compounds are collectively termed as cyanotoxins. There are 5 categories of toxins. First, hepatotoxins. In this group are microcystin, nodularins and cylindrospermins. The second groups are neurotoxins, and third group are dermatotoxins, and the fourth group are cerebral toxins. The different cyanotoxin molecules and their adverse health effects are being discussed. Microcystin, which is produced by Microcystis algae, causes abdominal pain, swelling of the liver, hemorrhagic stroke, failure of the liver, fatality, pneumonia and eczema. In long term, it may enhance the probability of cancer and liver failure. Another algal toxin is nodularin. Nodularin is produced by an algae, Nodularia spumigens. It is related to microcystin toxin. Another cyanotoxin is cylindrospermocin, which is produced by the alga Cylindrospermopsis. It causes abdominal pain, liver distension, liver and internal bleeding, pneumonia and eczema. Saxitoxin is produced by Anabaena lyngbya. It causes stinging, blazing, lack of sensation, sleeplessness, etc. and other fatalities. Anatoxin is produced also by Anabaena and lyngbya cylindrospermopsis. It also has similar adverse effect on the health, and it may also cause heart block and fatality. There are certain lipopolysaccharides produced by Oscillatoria species which are known to produce eczema and abdominal pain. So, these are some of the algal toxins or cyanotoxins which cause numerous adverse effect on the human and consumer health. There are another group of algae which are known as toxic diatoms. Toxic diatoms or Bacillariophyta, these are single celled photosynthetic organism and they cause contamination of seafood which results in intestinal cramps, nausea, diarrhea, migraine and irreversible loss of memory on short term and unconsciousness. It is essential to know what are the preventive steps and how we can control the algal bloom related illness. There are several measures that could be used successfully to control the algal bloom. Algal management strategy, catchment management strategy are 2 steps. Managing of algal blooms in stored water like dams and lakes by artificial destratification, biomanipulation, use of algistat and algicides, treatment of water and different treatment strategies like use of ultraviolet radiation, low chlorine doses, ozone, simazine products and cupricide usage are some of the ways by which we can control the growth of the algal bloom and algotoxins or cyanotoxin production. An integration of diverse methods will give better results than a single kind of treatment. The algal poisons as well as the fungal poisons, there are many legislations proposed by the government. In India, the protecting legislations for the safety of food are Prevention of Food Adulteration Act 1954 that has been abolished as Food Safety and Standards Act 2006. The Edible Oil Packaging Regulation Order passed in 1998, the Milk and Milk Product Order 1992, the Meat Food Product Order 1973, the Solvent Extracted Oil, De-oiled Meal and Edible Flour Control Order 1967, Essential Commodity Act 1955 and Vegetable Oil Product Control Order 1947. So, these are some of the legislations which have been in force from time to time to check the presence of toxins of microbial, fungal and algal origin to be present in the various food items. So, let us summarize. Food safety is of significant concern in India, having widespread public health implications. The entire food should be harmless, devoid from contamination and spoilage in its progress from its source till it arrives to the consumer. On the contrary, contamination of agricultural products and food by a variety of toxic molds and algae is very difficult to be controlled, as the food gets contaminated from water, fecal, air, soil, milking equipments, plant surfaces, infected animals and also by adopting unhygienic practices. Pathogenic fungi are identified to generate several kinds of lethal secondary metabolites which are not essential for the growth and are produced during the late exponential phase. Although neither the entire mold are pathogens nor are all the secondary metabolites, most fungal toxins are produced by some of the prominent fungus like Aspergillus, Fusarium and Penicillium. Moreover, some more toxic molds are there which may contaminate the food at different stages, and these include Alternaria, Acrimonium, Claviceps, Chaetomium, Cladosporium, Myrothecium, Rhizopus and Rhizoctonia. These fungi are either grouped as storage fungi or as field fungi, and they can infect the crop at different stages. Similarly, the marine products are also contaminated with the algal toxins, and it is essential to control and check the growth of these toxins in the food product for the overall well-being and health of the consumer. Thank you.